Hi, everyone. Today, I will talk about lipoproteins. Remember, in our previous discussion, we talked about lipoprotein as part of an example or a subsection of complex lipid, where we said complex lipid can be phospholipid. They can also be glycolipid, and they can be lipoproteins, where we said in lipoproteins, you have a complex of a lipid with a protein. These lipoproteins basically serve as the transport vesicles for lipids in the biological system, usually from one organ to another. This is based on the fact that we know that lipids are not soluble in water. And of course, if they are not soluble in water, the extracellular fluids, the blood, is more hydrophilic. Therefore, it is very difficult for lipids to easily be transported in these extracellular fluids. Therefore, there is a need for a means where these molecules can be transported from one part of the body to another, from one organ to another. So these lipoproteins, they form a complex of lipid and a particular type of protein called apoprotein. These proteins, before we talk about the structure of lipoproteins, or earlier, let me just give this explanation. You know that we made mention that these proteins, these lipids can be produced in the body. As well, you can also eat food that contain these lipids. So when you eat food that contain fat and oil, of course, after hydrolysis in the stomach before absorption, immediately inside the intestine, this lipid will, or the fatty acids and the glycerol will be utilized again to synthesize triacylglycerol, which is the neutral lipids, mostly. So the neutral lipids, the cholesterol, and some others will need to be packed and be transported after being taken to the systemic circulation. Then they have to be transported to different parts of the body for utilization in the body. How are these things transported? So also, we know that the liver also is involved in the synthesis of these fatty acids. So if the liver synthesizes fatty acids packed in form of triacylglycerol, how are these substances being transported to different parts of the body? So they need a vehicle, and that vehicle is these lipoproteins, where some lipids, most especially the water-soluble lipids, will now combine with some proteins called lipoproteins to form a macromolecular complex. This macromolecular complex called lipoproteins, they are the ones responsible in transporting the lipids to different parts of the body. We are going to see the structure of these lipoproteins. But before then, these lipoproteins were classified into four. We said we have chylomicrons, low-density lipoproteins, very low-density lipoproteins, high-density lipoproteins. And this is based on their mobility in an electric field. In an electric field, you can see based on size, you can see the high-density lipoproteins is very small, therefore it can easily migrate. And you have chylomicrons, which is bigger, and it is very difficult to migrate slowly. Based on structure, like we said, these lipoproteins are made up of a proteins and the lipids. This particular protein is called apoproteins, while the lipids can be neutral lipids, they can be phospholipids, and any other type of lipids. How is the structure formed? The structure of the lipoprotein composed of an outer shell, which is called a coat, and an inner core. You can see from the first structure by the left here, you can see an outer part. And this outer part is made up of usually water or amphipathic lipids. You know, example of amphipathic lipid, we say phospholipid. We know phospholipid have the hydrophobic and the hydrophilic region. And we know cholesterol also because of the OH group. That place with OH group is a bit hydrophilic in nature. They can interact with the water. So this type of lipid can now form this kind of a layer. So when they form this layer together with proteins, then the inside the core, you have some neutral lipids that are highly hydrophobic. They do not interact. And as well as cholesterol esters. So cholesterol esters and triacylglycerols are usually the main lipids that are found inside. When you look at another stroke, the other